So for filtration, we want to see kind of how it happens. So what we have is when we look at Bowman's capsule, we're going to see that the capsule is lined with simple squamous epithelia. And then when you look at the glomerulus is also simple squamous epithelia. Now, I'm not going to go into detail about all of these structures that are associated with the epithelia. What I basically want you to know is that we've got simple squamous epithelia both with the capsule and with the glomerulus, and things are going to diffuse out of the glomerulus, and things can diffuse into the glomerulus as well, in and out of the capillaries. Most of the material is going to be diffusing out of the glomerulus and forming this filtrate. And we'll look at the pressures that dictate that process. But what I want you to keep in mind is, in this area where it's simple squamous epithelia, we aren't going to have channels, we're not going to have pumps that are in the membrane of these cells. Whatever is able to diffuse in or out of the blood is going to do so by going through these filtration slits. They're basically little gaps that are between the epithelial cells. Again, these are capillaries, and we know that in capillaries things can diffuse across those cells very easily because they are simple squamous epithelia. The glomerulus is a particular type of capillary where it has very large diameter pores in it. And with these pores and these slits, that's going to allow material to be able to diffuse across the glomerulus without having to go through channels and requiring pumps. So pretty much anything that is small enough is going to be able to leave the glomerulus. And that's going to be mostly due to blood pressure. We're going to see that in the glomerulus, blood pressure is particularly high there, higher than it is in other capillaries throughout the body. And we'll talk about why that is the case. But the fact that the pressure is high in the glomerulus is going to help to force material out of the blood and then form this filtrate. So we're going to get lots of things that come out. We're going to get water that comes out of the blood plasma. We're going to get things like glucose, fatty acids, metabolic waste, ions. Anything that's small enough to fit through those slits or fit through those pores is going to come out. Now, let me just also say, things that are larger, like plasma proteins, cells, red blood cells, white blood cells, they're not going to be able to fit through those pores or those slits. So they're going to get left behind. They're going to end up staying in those capillaries and being a part of the blood. Now, this is good because we want things like metabolic waste to come out of the blood and into this filtrate so that we can eliminate that from the body as urine. The problem is we're also getting a lot of things that we don't want to get rid of coming out and ending up in that filtrate, like glucose, like fatty acids, like amino acids, and some ions. Things that we actually want to keep because we spent all this time in the digestive tract breaking material down to get all these great nutrients. So we don't want to get rid of them. The body will either utilize these things right away, or we saw that the body will store these things or turn it into something else that it can store, like turn it into fats so that we can store those energy reserves and use them later. So we don't want to get rid of this stuff, but it's going to come out, and it's going to come out because it's small enough and the blood pressure is forcing it to come out. So that means once that filtrate gets into the tubule, then we're going to have to reabsorb most of that material. We're going to have to take it from the tubule and put it back into the blood. back into the peritubular capillaries so that we can keep those good things. And that's what reabsorption does. It allows us to get that material back. Now, we can also, we said, do secretion. And what secretion is doing is, if we didn't get all of the metabolic waste out here during filtration, or maybe the body says, oh, our blood calcium levels are low. We actually need to take some of that, our calcium levels are high. We actually need to put more calcium into the urine. Or maybe we need to put more sodium into the urine. Or maybe put more water into the urine. Secretion allows us to do that. So whatever didn't end up in the filtrate here at the corpuscle, secretion is going to allow us one more chance to take things out of the blood and put it into the filtrate and become urine before it leaves the kidney, and then we can't adjust it anymore. Okay, are we good with that? Okay, great. So once we leave the corpuscle, then filtration is done. Okay, again, filtration happens at the corpuscle. Once we then move over to the first part of the tubule, the PCT, this is where we're going to do most of our reabsorption. Now, we can do reabsorption throughout the whole tubule, whether it's the PCT, loop of Henle, DCT, even in the collecting ducts. But most of it is going to happen right away here in the PCT. That's where we're going to reabsorb most of the material. And exactly what we absorb here is going to depend on exactly what kind of channels are present or what kind of pumps are present here. So we're going to see that we're going to absorb a lot of organic nutrients, which means that there's going to be a lot of glucose pumps here, amino acid pumps. We're going to absorb a lot of ions, meaning that we have a lot of sodium channels here and chloride channels and potassium channels. We're going to be able to absorb water. Water can either go right through the membrane of these simple cuboidal epithelial cells, which you see have microvilli on their apical surface, and that should make sense. Microvilli is there to increase surface area. Why do we want to increase surface area? Because we're reabsorbing a lot of material. So let's make that surface area larger. Okay. Now, when we do reabsorb this material, remember that it's going to go, let's see, if we're in, so if this is the PCT, and again, it would be lined with simple cuboidal epithelia. So that would be going like this. Right, all the way down, the PCT. And the surface has microvilli, so all these guys have microvilli on their surface. So if we're reabsorbing material that's here in this filtrate, that's traveling through the PCT, then that means that whatever we're reabsorbing is going to go across this apical surface of the simple cuboidal epithelial cells. Then it will go across the basal surface of the simple cuboidal epithelial cells and end up out here in the extracellular environment, this extracellular fluid. And that's referred to as the interstitial fluid. Then, whatever we're reabsorbing, let's say it's sodium. So let's say sodium goes across the apical surface, then sodium goes across the basal surface, then sodium's out here in the interstitial space. Then it's going to diffuse into the peritubular capillaries. That again would be surrounding this PCT. So you can see that sodium has to go across several plasma membrane areas. And again, it can do that because there are channels there or there are pumps there that are allowing that particular compound to diffuse. if there are no channels or pumps, then that particular compound can't get reabsorbed. So when I talk about reabsorbing and secretion, I'm going to say that it goes from the filtrate into the peritubular capillaries or into the blood. Just remember that it really is going from the tube into the extracellular space or interstitial fluid and then into the blood. Just like any time things are diffusing, they're going through that space. Okay, then we're going to go into the loop of Henle, and we're going to see that the loop of Henle has a descending limb. So as it's descending, the fluid is moving towards the renal pelvis, and then it's going to go into the ascending limb, and that fluid's going to be moving up towards the cortex again. Now, we're going to see that each of these limbs is going to contain a thick segment and a thin segment. And those segments will have a different type of epithelia. The thick will have a cuboidal epithelia. The thin segment will have a squamous epithelia. And I'm not really concerned that you know that so much. What I want you to know is that these different parts of the limbs are going to be selective for certain types of compounds. So what we're going to see is in the thick segment, this cuboidal epithelia, we're going to see that there are pumps in there. Pumps for sodium and pumps for chloride. And we're going to be able to pump sodium and chloride out of the tubular fluid, which would be here, the filtrate. We're going to be able to pump it out of the tubular fluid and into our peritubular capillaries. So that means we would be reabsorbing sodium and chloride, taking it out of the filtrate, putting it back in the blood. And these are going to be active transport pumps. They're going to be actively pumping sodium and chloride. So the loop of Henle is very specific for what it's going to reabsorb. And so we're going to reabsorb sodium and chloride, and wherever sodium goes, water will follow. So water is also going to get reabsorbed here at the loop of Henle. We're going to see that the thin segment of these limbs is freely permeable to water, but not to any of the other solutes. Now that's going to be important when we look and see exactly how is water and sodium and chloride getting reabsorbed. We're going to see that because different parts of the loop of Henle are permeable to some things but not others, it's going to set up various gradients, concentration gradients. And again, I think we'll talk about this in more detail, but just think if we wanted to reabsorb water that was here in the filtrate. Water is not going to leave this filtrate unless it has a reason to. Water will move via osmosis. So if we want water to move like this and ultimately into these peritubular capillaries, we have to have a certain osmotic concentration out here in that interstitial fluid to draw that water out. So if we want water to come out here, the osmotic concentration out here has to be high. And that will then cause water to move from a high water concentration to a lower water concentration. And we'll talk about that more. But that's what's going to be going on at the loop of Henle. We're going to also be reabsorbing, in particular, we're going to be reabsorbing sodium, chloride, and water. Now, when we get to the DCT, okay, this is going to be a slightly smaller diameter than the PCT. It also is lined with epithelia, simple cuboidal epithelia, but here we're not going to have any microvilli. And we're going to see that in the DCT, we've got kind of three important things happening. We also are going to reabsorb here in the DCT. We're going to have what's called selective reabsorption of sodium and calcium. Now the reason that selective is there is because in the DCT, things are going to get reabsorbed. Sodium and chloride are going to get reabsorbed based on what hormone levels are. So if we have a high amount of PTH in the blood, that's going to cause more calcium to get reabsorbed. If our calcitonin levels are high, then we will reabsorb less calcium here at the DCT. So the DCT is going to be under the influence of hormones, and that's why it says selective reabsorption. We can also selectively reabsorb water here at the DCT, and that's going to be under the influence again of hormones, ADH, and then also aldosterone, and we'll talk about how that works later. So in the PCT and then the loop of Henle, there are always a certain number of pumps or channels there. And so we pretty much are always going to be absorbing or reabsorbing a certain amount of glucose or amino acids or ions or water, because there are always a certain number of pumps there. The DCT, though, is different. We can change how much of this material we're reabsorbing because it's under the influence of hormones. So we can change the pump numbers. We can either have more calcium pumps in the membrane or less, and that will then cause more or less calcium to get reabsorbed. So that is the major difference. DCT is under the influence of hormones. That's where we can change how much we're reabsorbing based on what the body needs at that particular point in time. Now, we can also secrete in the PCT and the loop of Henle, but we're going to do a lot of secretion in the DCT. So we said the PCT is where we do most of our reabsorption. Then I would say that the DCT is probably where we're going to do most of the secretion, even though we can do it in other places. And the types of things that we would be secreting are going to be under the influence of active transport. We're going to be actively taking ions, acids, drugs, toxins, various metabolites. Let's say you took some Tylenol, and the liver breaks down that Tylenol, releases a variety of metabolites. The kidneys then will secrete those metabolites. So if those metabolites, those drugs, those toxins didn't end up in the filtrate, then we have pumps that will actively take these various compounds from the blood, transport them into the interstitial fluid, and then transport them into our tubule. And again, a lot of that happens at the DCT because that's where you tend to find a lot of these very variety pumps for different types of drugs and toxins and other things. Now, the collecting system. This is, again, what the DCTs are draining into, the collecting duct or the collecting system. The epithelia is typically columnar, simple columnar epithelia. And this is going to be the last place that we can adjust that filtrate before it becomes urine and leaves the kidney. So the collecting duct is where we make the final tweaks over osmotic concentration, how much calcium is going to be in that filtrate, how many hydrogen ions are going to be in that filtrate, how much water. So this is the last point where we can tweak things, and once that filtrate leaves the collecting ducts, then it'll go through the renal papillae, then the minor calyx, major calyx, renal pelvis, ureter, bladder, urethra. Once it leaves the collecting ducts, we can't change it anymore. Regardless of what hormones are in our blood, we can't change it. Okay? All right. So, when we talk about various metabolic wastes and urine production, again, the goal of producing this urine is to get various waste products out of our blood, out of our body, and to maintain homeostasis of our pH, of our blood volume, and of our blood pressure. So we're going to see that initially, when we filter the blood, when we do filtration at the renal corpuscle, that filtrate is going to look very much like blood plasma, because that's what really that's what it is. So it's going to be mostly water with some things dissolved in it. So it's going to look very similar to blood plasma. But as that filtrate travels through the tubule, through the PCT, the loop of Henle, the DCT, and collecting duct, it's going to start to look less and less like blood plasma and more like urine. And the main difference is urine is going to be far more concentrated than blood plasma is. It's going to have about 4 times the osmotic concentration of blood plasma. And we've got to keep that urine nice and concentrated because we don't want that water to end up in the urine. We want to keep most of that water for ourselves. We want to put it back into the blood. So even though we are going to get rid of a fair amount of water, that urine is going to be much more concentrated than our blood plasma is, than our extracellular fluids are. It's also going to tend to be, urine is going to tend to be on the acidic side. Now, urine will range, can go from a pH of 4 up to, like a pH of, I think it can go up to like 8 or so. And I think the normal urine pH is around 6 or something like that. But when you look at our cells and the various chemical reactions that our cells do, many of the waste products are acids. So usually what we're concerned with is our blood pH becoming too acidic. Usually we're not worried about it becoming too basic because, again, our cells are producing a lot of acid byproducts. So what that means then is if we want to keep our pH constant, 7.35 to 7.45, we've got to get rid of a lot of those hydrogen ions. And we saw that we can do that through the respiratory system by changing our CO2 levels. But another way is at the kidneys, we can take those hydrogen ions that are in the blood and we can put them in the urine. And so that's why urine tends to be on the acidic side of things, because we're taking those hydrogen ions from the blood, putting them in the urine, so that our blood pH stays constant. Now, there are 3 metabolic wastes that are most common in the urine, and so you should be familiar with those. These are all nitrogen-based compounds, and our body just is not very good at dealing with compounds that have nitrogen in them. Plants do very well with nitrogen-based compounds, but our body doesn't. Our body tends to prefer carbons and hydrogens and oxygens, maybe some sulfates here and there. We do obviously have nitrogens in our body. Our DNA has nitrogen in it, our RNA does, some of our amino acids, but a lot of these wastes we just can't deal with. So the major ones are urea, and we get urea by breaking down amino acids. So anytime that our body takes an amino acid and throws it into the mitochondria to make ATP, we're going to get urea as a byproduct. Or our liver can take one amino acid and break it down and turn it into another amino acid. Or our liver can take an amino acid and break it down and turn it into glucose or into fats. So anytime that happens, urea is produced, and we got to get rid of it. Creatine. We talked about this with skeletal muscles. And skeletal muscles, they can take creatine and ATP, take a phosphate off of that ATP and make CP, creatine phosphate. Now, when our skeletal muscle cells use that CP, then they're going to generate ATP and creatine back again. And the cell has to get rid of that creatine. And so this is also going to be another byproduct. So when your skeletal muscles are really active, particularly if you're doing activities that are high intensity and short in duration, like sprinting, powerlifting, then that's when you tend to use CP a lot. And again, breaking that down to get quick ATP, that's your waste product you've got to get rid of. And then uric acid. Uric acid is formed when you recycle nitrogenous bases from RNA. So this could be formed in a variety of ways, but anytime you're using RNA, breaking down RNA, then uric acid. So these are the common nitrogen-based metabolic waste products that you see in the urine. And if they're worried about how well are your kidneys functioning or worried about various metabolic issues, they will test and see, are these at normal levels, or are they too high, or are they too low? So, back to these major processes. Again, we've already said filtration, reabsorption, secretion, and now what we're going to do is look in more detail at each of these processes, and then talk about various factors involved in these processes. So again, for filtration, remember that this happens at the renal corpuscle between Bowman's capsule and the glomerulus. Here we're going to produce filtration based on pressure, blood pressure, forcing material out of the glomerulus. Whatever is small enough to pass through those slits will end up in the filtrate. For reabsorption, this is taking water and solutes from the filtrate, putting it back into the blood. That's going to be back into the peritubular capillaries. This is going to be based on simple diffusion, if we're talking about fats just diffusing across the membrane, channel-mediated diffusion, carrier proteins, if we're talking about compounds that are water-soluble. They're going to need a channel or a carrier to get across the apical surface of the membrane, the basal surface of the membrane, and then into the blood. Okay. Water will tend to follow passively via osmosis, again based on concentration gradients. Make the extracellular environment osmotically high in terms of concentration, and water will come out of the tubule via osmosis. Secretion, again, is taking material that's in the peritubular capillaries and putting it into the tubule, making it part of the filtrate. And again, this would happen if we didn't get everything into the filtrate initially. So the movement of filtration and secretion are the same direction. The movement of absorption is in the opposite direction. So filtration and secretion from the blood into the filtrate. Reabsorption is opposite, from the filtrate back to the blood. Okay, so we're going to start with filtration, and we're going to say, what are these pressures? What are these forces that are causing a filtrate to be formed? So why does the fluid leave the blood? Why does the fluid and whatever is dissolved in the fluid, why does it leave the blood? What's forcing it to leave the blood? Why doesn't it just stay in the blood? And when we look at what causes filtration, there are going to be two major pressures that are involved. One's going to be hydrostatic pressures, and then the other one is going to be osmotic pressures. That's what's going to cause this water and whatever's dissolved in it to move across the glomerulus. So first of all, we want to look at these hydrostatic pressures. And we said that when we look at Bowman's capsule, okay, and the glomerulus is inside, so I'm just going to draw a little piece of the glomerulus. So this is the glomerulus. This is Bowman's capsule. And we said coming into the glomerulus, we have the afferent arteriole comes into it. And leaving the glomerulus, we have the efferent arteriole. Now, again, this is different from other capillaries that we've looked at. Other capillaries have an arteriole coming into the capillary and then a venule leaving. But here we have two arterioles. And these arterioles, just at rest, what you're going to see is the afferent arteriole is going to have a slightly larger diameter than the efferent arteriole. And what that does is, if you think about it, blood comes in through the afferent into the glomerulus, and then we decrease the diameter of the efferent a little bit. So that makes it harder for the blood to leave. It can't come in, it can't exit as easily as it came in because we decrease the diameter a little bit. So what that does then is it makes the pressure here in the glomerulus a little bit higher than your average capillary. And we're going to see that we're going to be able to change the diameter of the afferent and efferent to try to keep the pressure here in the glomerulus constant. And we'll talk about all that in a minute. Now, this pressure here, basically this blood pressure that's inside the glomerulus, is our glomerular hydrostatic pressure. And that pressure tends to force water and solutes out of the glomerulus and into the space of Bowman's capsule. That's our glomerular hydrostatic pressure. The fact that there's high blood pressure in there. So the glomerular hydrostatic pressure is in favor of filtration, in favor of producing a filtrate. Then, because the water and solutes aren't leaving the glomerulus and just going out into the interstitial space, because that water and solutes is coming out into a confined space, a capsule, the capsule itself is going to exert pressure on the water and solutes. And that's called the capsule hydrostatic pressure. And the pressure exerted by the capsule is tending to force water and solutes right back into the glomerulus. So that's your capsule hydrostatic pressure. So the pressure exerted by the capsule tends to oppose filtration because it tends to push that stuff right back into the glomerulus. So what you want to do is you want to add up those two forces, and that will give you your net hydrostatic pressure.
It means subtracting, right?
Add up those two forces. And since they're opposing forces, you're subtracting one from the other. But you basically are adding up all the forces.
Oh, I see.
This is a positive force, that's a negative
I
force,
see what
and
you're
you
saying.
add them together. So your net hydrostatic pressure is your glomerular hydrostatic pressure, and your capsule hydrostatic pressure is opposing glomerular hydrostatic pressure. And if you look at real numbers, if you look at the blood pressure here in the glomerulus, again, it's higher than normal. It's around 50 millimeters of mercury. That's a little bit high for a capillary. You think the pressure in your superior and inferior vena cavas is usually about 2 millimeters of mercury. Pressure in your aorta during ventricular systole is around 120 or so, something like that. This is fairly high for a capillary. But that's your glomerular hydrostatic pressure. And then the pressure exerted by the capsule is about 15 millimeters of mercury. Not a lot, very little. But when you add up those two forces, it gives you a net hydrostatic pressure of 35 millimeters of mercury. So what that number is telling you is the net effect of these hydrostatic pressures is for water and solutes to come out of the glomerulus and into this space and to form a filtrate. It's not a huge pressure. It's only total 35 millimeters of mercury that's essentially forcing this water and solutes out. But it's enough to form a filtrate, right? And that's a good thing because we want to form a filtrate, right? Why do we want to form a filtrate? We want to form what? Urine, right? And why do we want to form urine? Eliminate waste, okay? So this is good. We want to form a filtrate. So now we have to look at what's called our colloidal osmotic pressure. And what this is, so when we have our hydrostatic pressures forcing water and solutes out of the glomerulus, we have to think, what's left here in the glomerulus? What's left in there, in the blood? So we've got red blood cells, we've got white blood cells, and we have anything that's too big to leave, which would be our plasma proteins. And as water and solutes leave, and we're left with just a little bit of fluid and cells and these big plasma proteins, that makes this blood very concentrated, has a very high osmotic value. And whenever water is in an area where it's by a high osmotic concentration, that draws water into the area. Right? If there's a lot of stuff here in the blood, that means there's very little water in the blood. And so now you've got high water concentration out here in the capsule, low amount of water here in the glomerulus, and water will move back in via osmosis. Okay? So that's our blood colloidal osmotic pressure. The fact that we have plasma proteins in the blood sets up an osmotic situation that's going to draw water back into the blood, okay? Again, via osmosis. Now, this is also why you don't want to see plasma proteins in your urine, because if you've got plasma proteins in your urine, it's saying something's going wrong here, and those plasma proteins are coming out into the filtrate and ending up in the urine. And what that's going to do is it's going to make it harder for water to be able to go back into the blood, which means that you may be starting to lose too much water in your urine because you can't draw that water back into the blood.
I don't know if this is related or not, but what exactly is a kidney stone? Does it have anything to do with this kind of?
A kidney stone is usually when you have, it's usually calcium. If you have really high calcium levels, you can get things starting to kind of conglomerate and fall out of solution. And then that literally will put, you've got a little particle that gets stuck in those tubes. And so then the filtrate can't flow through the tube. But it wouldn't be necessarily due to this right here. Okay, so the colloidal osmotic pressure is going to oppose filtration. Again, because it's tending to draw this water that just came out, it's tending to draw it right back into the glomerulus. So what we want to do is we want to add up these two forces to get our filtration pressure. And our filtration pressure is equal to what we just calculated, our net hydrostatic pressure minus the blood colloidal osmotic pressure, because that opposes filtration. So that's why we're subtracting it. And if you look at real numbers, we said our net hydrostatic pressure is 35 millimeters of mercury. And the osmotic force that's tending to draw water and solutes back into the glomerulus because of osmotic pressure, that tends to be about 25 millimeters of mercury. And so when you add these up, you have 10 millimeters of mercury is your filtration pressure. So again, what this number is telling you is when you add up all of these different forces, the net effect is water and solutes will leave the glomerulus, not all of it, obviously, but some water, some solutes are going to leave the glomerulus and form a filtrate. But the overall pressure that is causing that water and solutes to leave the glomerulus and form a filtrate, it's not very big, 10 millimeters of mercury. So now the issue is we have to make sure that we have everything just right. We've got to make sure that our blood pressure doesn't get too high or too low. We need to make sure that we're forming enough plasma proteins that actually stay in the blood that help to draw that water back. Everything has to be just right so that we end up with a net positive number and we form a filtrate. So that's going to take us into this next number. This is the number we're really concerned with. And this is called our glomerular filtration rate. So this is how much filtrate are each of the kidneys producing every minute. And that's about 125 mils per minute. And that's actually a lot if you think about it. So that's the number that we're concerned with, the glomerular filtration rate. We don't want that number to become too high. We don't want that number to become too low. If that number were to become too low, we could get to a point where we weren't making a filtrate at all, which means that we weren't forming a urine, which means that all of those toxins would end up staying in the blood and damaging any cell that they come in contact with as they're traveling through the blood. That means that we wouldn't be able to regulate blood volume, we wouldn't be able to regulate blood pressure, at least through the urinary system. We would have a hard time regulating blood pH. So we don't want that number to become zero. I absolutely don't. We also don't want it to become too high, because then that says we're putting too much water into the urine, and we could start to get dehydrated if that happens. So if you look over the course of a single day, if we're making 125 mils per minute, and that means over a single day, the glomeruli are generating about 180 liters or 48 gallons of filtrate every day. Now, that's a lot, right? Are you urinating out 180 liters of urine for 48 gallons? No, you're not. And that's because most of the filtrate that we just made, 99% of it is going to get reabsorbed as it enters into the tubules. So only 1% of what we make is going to end up becoming the urine. So we're going to reabsorb almost all of that material. Now again, this glomerular filtration rate depends on these filtration pressures. And if we would have our blood pressure, for example, if our blood pressure there at the glomerulus, which we said is about 50 millimeters of mercury, if it were to drop by just 20%, then this number, so if that dropped by 20%, that would go from 50 down to 40, right? So if that number were 40, 40 minus 15 would give us 25 here, and then we would have 25 minus 25, that would give us 0. No filtration being produced, no filtrate being produced. So that blood pressure in the glomerulus goes from 50 to 40, which again, that's not a big difference. If it drops by 20%, then we can get to the point where we're not producing urine at all. And this happens if you're really stressed, this can happen. If you're in sympathetic mode for too long, this can happen. Sometimes if you have a severe injury, this can happen. So maintaining blood pressure here at the glomerulus is really critical. Obviously, maintaining blood pressure throughout the body is important because that influences blood flow throughout our blood vessels. But we also need to make sure right here at the glomerulus, that pressure stays relatively constant.
How can they calculate the GFR?
They probably put some kind of dye into the blood, and then they monitor those particles as they're coming out. I'm guessing. I'm sure there's a few different ways to do it, but that's probably one way. So hemorrhaging, shock, dehydration, they're all conditions that can cause GFR to drop. So what we want to say is, how do we control glomerular filtration rate? Since it's so important, how do we control it? How do we make sure that we keep producing filtrate at about 125 mils per minute? Now, that means that we're going to be talking about homeostatic mechanisms, right? Keeping something constant. And every other time that we've talked about homeostatic mechanisms of various systems, whether it's heart rate or blood pressure or stroke volume or respiratory rate, we've used the nervous system and various neural reflexes, receptor being activated, send it to a control center, process the information, change, increase or decrease sympathetic versus parasympathetic activity. And that will happen here, but it's not the primary way that the urinary system maintains GFR. The urinary system primarily maintains GFR through autoregulation at a local level, meaning that every single one of those 1.5 million or so glomeruli in the kidney are dealing with their own GFR themselves. They're making sure that their GFR is constant, and then the glomeruli beside them are making sure that theirs is constant, and so on and so forth. And this is constantly being tweaked. Each glomerulus is constantly tweaking different things to make sure that their GFR is constant. And we're going to look and see how that happens. So that's autoregulation at a local level. When we've talked about homeostatic mechanisms at the local level, that means we don't involve the nervous system, we don't involve the endocrine system or hormones. Those are things that are going to influence systemic responses. So this is generally how the kidneys deal with GFR. On a local level, each glomerulus doing its own thing. Now, if it can't do that, or if other things start to happen, then hormones will kick in, and hormones will start to influence different things, and we'll talk about those things, that keep GFR constant. And then the nervous system can play a role, but hormones and the nervous system are not going to be the first go-to scenarios, like we saw with regulating blood pressure throughout the whole body, or regulating oxygen levels throughout the whole body. The kidneys will do most of it themselves at the local level. So how do they do that? What are the kidneys doing? And really, what's the major thing that the kidneys are doing constantly is these arterioles lined with smooth muscle are going to respond, either contract a little bit more or relax a little bit more based on what's going on here with the GFR, with the glomerular filtration rate at that particular glomerulus. So we said that we can dilate or constrict these arterioles. And we said at rest, the efferent arteriole is a slightly smaller diameter than the afferent. Okay? So if we were to have, let's say that we were to have a drop, so let's do this. If the glomerular hydrostatic pressure, the GHP, if that started to drop in this particular glomerulus, then that would cause more or less filtrate to be produced because of this. Not homeostatically, but because of this. Less, right? And that would ultimately cause our glomerular filtration rate to drop, and we don't want that to happen. So if glomerular hydrostatic pressure drops, if our glomerular filtration rate is dropping, we want to get those numbers back up so that we keep forming a filtrate. So if the pressure in the glomerulus is starting to drop and filtration is starting to drop, would we want to dilate or constrict this afferent arteriole? We would want to dilate the afferent arteriole, right? Dilating this is going to allow blood to flow into this capillary bed faster, right? Same thing like at every capillary bed. If we want blood to flow into that bed faster, we dilate the afferent arteriole. It's like opening up the floodgates so more blood can come in. Okay? More blood coming in means the pressure in here is going to start to increase. Now, what would we want to do to the efferent arteriole? We want to constrict that. Constrict the efferent. So if the pressure in the glomerulus starts to drop, which puts us at risk for decreasing our glomerular filtration rate, then we can have either or of these things happening. Okay? The afferent arteriole will dilate, which allows blood to come into the glomerulus easier, and then we're going to decrease the diameter of the efferent arteriole, make it harder for that blood to leave. If the blood can't leave as easily, then the blood's going to be here in the glomerulus just a little bit longer and cause the pressure in there to start to rise. Yeah.
Did you say you're going to do one or the other? Or
You can do one or the other or both together. Yeah. And it really just depends on how severe the changes is. And again, your glomerulus, every single glomerulus and every single afferent and efferent arteriole are constantly changing. They're constantly either constricting a little bit more or dilating a little bit more to keep that number in there constant. Okay? And this is happening 24/7 constant. Every single one of those is constantly doing that. So the opposite would be true here. If the pressure in here started to become too high, which means our glomerular filtration rate would start to become too high, then we can constrict the afferent, make it harder for blood to come in, and we can dilate the efferent, make it easier for blood to leave, and that will cause the pressure in here to start to drop. Put less blood in here and make that blood easier to leave. Okay, so we do just the opposite. Okay. Now, also what will happen, we've talked about the fact that if smooth muscle cells stretch, they will respond by contracting. That's an inherent property of these cells. So if we start to have an increase in systemic blood pressure, so now we're talking about not just at one glomerulus. Okay? If across the whole body we start to see a rise in systemic blood pressure, then what that means is at all of these glomeruli, chances are the pressure will be a little bit higher in all of them. And if the pressure is a little bit higher coming into the glomerulus, that will cause the afferent arteriole to start to stretch a little bit, which will then cause it to contract, decreasing the diameter. And as we decrease the diameter here, the pressure inside will start to drop. So again, autoregulation is dealing with every single individual glomerulus, but it also, autoregulation can deal with if we have a systemic problem. Pressure throughout the whole body is increasing, causing every single glomeruli to increase in pressure. Then autoregulation can also deal with that, deal with every individual glomerulus. Okay. questions on that?
People have the kidney problem, like dialysis. That means glomerular, or it's not actually.
Yeah, so people that have to go on dialysis have to do that because of it could be lots of different things wrong with the kidney. But ultimately, people that are on dialysis because they can't produce a filtrate adequately. This number is too low, and that could be for many, many reasons. So the machine basically does it for you. You get hooked up to a machine, and instead of your blood flowing through here to get filtered, your blood then goes through a machine and gets filtered, and then that clean blood goes back into your systemic circulation. Yeah.
So it's more than living with one kidney, with the behavior of that one kidney
You just have to, because it'll still do the same thing. Everything will be exactly the same. You're just asking that kidney to do more, to filter twice as much blood as it normally would. So it does exactly the same thing. You just want to be extra careful because you're asking it to do more work. But all the properties would be exactly the same.
Okay, so even though there's more blood,
It would be
The
producing
pressure-
filtrate faster, but the pressure will be the same. Yeah. I shouldn't say it's producing faster. It's producing overall more, let's see, that one kidney would be producing a greater amount of filtrate, what two kidneys would normally do, the one is doing. But in terms of pressures, those are going to stay relatively constant. Okay. So, if we look at hormonal regulation, how can hormones affect the GFR? And why would we want hormones to affect the GFR? There's a few different hormones that can influence the GFR. One is called the renin-angiotensin system. And what this is if we have a drop in GFR, and again, think about a decrease in pressure in the glomerulus would cause a drop in GFR. That will actually cause a group of cells that we skimmed over, and let me just go back to a few slides here. Let's see, where was it? Okay. In the DCT, there is a little structure associated with the DCT called the juxtaglomerular complex. And this structure here is actually an endocrine structure associated with the DCT. And this endocrine structure secretes hormones like EPO and TPO. Remember, EPO is erythropoietin. That hormone is released when oxygen levels throughout the body start to drop. EPO goes to the bone marrow and ramps up red blood cell production. Also releases TPO, which causes more platelets to be produced. And this structure releases an enzyme called renin. So, if we go back here, if we have a drop in glomerular filtration rate, and again, that could be due to something that's happening systemically, like a drop in blood pressure systemically, or it could be something that's happening just here within the kidneys itself. But whatever the reason, we have a drop in GFR. That will cause release of renin from that juxtaglomerular complex of the DCT. And renin will activate a chemical called angiotensin and cause it to form angiotensin II. Now, angiotensin II can go do a variety of things in the body. So I'm actually going to come over here to this figure because this is a nice summary slide. So what this is looking at is if we have a low systemic blood pressure, so throughout the whole body, blood pressure is dropping, and if blood pressure is dropping throughout the whole body, then that means the pressure here in the glomerulus, the GFP, would also drop, which would cause our filtration rate to drop. And we already talked about autoregulation, what will happen in terms of the change in diameter of the afferent efferent arterioles. In terms of hormones, a drop in GFR, and again, in this case, it's due to a drop in systemic blood pressure, causes renin to be released. Renin is an enzyme that causes the conversion of angiotensinogen into angiotensin II. Angiotensin II can do a variety of things in the body. One thing it can do is it can go to our various arterioles. Actually, it can go to, that should actually say peripheral arteries. It can go to various peripheral arteries and cause vasoconstriction. And we've already talked about the fact that if you vasoconstrict your larger peripheral arteries, that will increase your systemic blood pressure. And if you increase your systemic blood pressure, the pressure here in the glomerulus will also rise, and that will cause GFR to go back up to 125 mils a minute. Now, angiotensin II can also come over here to the adrenal cortex. Now, at the adrenal cortex, angiotensin II will cause the adrenal cortex to release aldosterone. Now, aldosterone hormone, a mineralocorticoid, enters the bloodstream, and it will come over to the kidney tubules. In particular, aldosterone will come to the DCT, and it will cause the DCT to make more sodium potassium ATPase pumps. It literally will make those simple cuboidal epithelial cells make more sodium potassium ATPase pumps and put them in the membrane. And if we have more sodium potassium ATPase pumps, it means that we will increase the amount of sodium that we're reabsorbing from the filtrate. As we reabsorb more sodium, put more sodium back into the blood, water will follow. It means we reabsorb more water. If we reabsorb more water, what is that going to do to our blood volume systemically? Increase our blood volume systemically. What does that do to our blood pressure systemically? Increases it. Increasing our blood pressure systemically will cause the glomerular hydrostatic pressure to rise, and that will get our GFR higher. Yeah.
So would that come more into play if your if the filtration rate was low for a long time?
Exactly, yeah. So autoregulation is doing most of the work on kind of a minute-by-minute basis. But if you start to have prolonged types of activities, then the hormones start to become bigger and bigger players. Now, also, the nervous system can play a role as well. So we said that if systemically blood pressure starts to drop, which division of the autonomic nervous system will start to become more active? Systemically, blood pressure is dropping. Which division of the autonomic nervous system is going to cause the heart rate to increase? Sympathetic. Which division of the autonomic nervous system is going to cause our peripheral larger blood vessels to vasoconstrict? Sympathetic. And if our heart rate is increasing, our stroke volume is increasing, our larger peripheral vessels are vasoconstricting, what does that do to our blood pressure systemically? Increase. And if our blood pressure systemically is increasing, then our glomerular hydrostatic pressure will increase, our filtration rate will increase, and we'll get back into homeostatic range. But again, this is only going to become, the autonomic nervous system is only going to become a big player in really extreme situations, okay? In terms of the kidney, So, for the most part, if our glomerular filtration rate drops, then autoregulation is going to tweak the diameter of the afferent and efferent arterioles. Systemically, if blood pressure is dropping, which then causes the pressure in here to drop and causes filtration rate to drop, then the autonomic nervous system can play a role systemically to cause the pressure in here to rise. And hormones can also play a role to cause the pressure in here to rise. Okay, now on this slide there are two other hormones, and these two other hormones, ANP and BNP, atrial natriuretic peptide released from the atria of the heart, BNP, brain natriuretic peptide, it's actually released from the ventricles of the heart. These two hormones are going to oppose aldosterone, what we just talked about. So aldosterone, we said, is released, let's put it right over here. Aldosterone is released if blood pressure systemically drops. And what aldosterone ultimately is going to do is cause more sodium to get reabsorbed, more water to get reabsorbed, increases blood volume, that increases blood pressure. ANP and BNP do just the opposite. So ANP and BNP are going to be released if the atria and the ventricles are stretching too much, which basically means if blood volume is too high, if we have too much water in our blood volume is too high, then the atria and the ventricles will stretch more. And that will then cause ANP and BNP to be released. And what they are going to do is they are going to decrease how much water gets reabsorbed so that we lower blood volume and thus we lower blood pressure. So ANP and BNP do the opposite to aldosterone and ADH, which we'll talk about later. Okay? ADH and aldosterone do similar things in terms of blood volume, blood pressure. They both, aldosterone and ADH, both are going to cause an increase in blood volume, thus an increase in blood pressure. ANP and BNP do the opposite. These guys are going to decrease blood volume and decrease blood pressure. Okay. All right, so we look at that. Okay. We just talked about that. Other things that ANP and BNP can do. Okay? So I said that ANP and BNP are going to be released if blood volume is high. And usually blood volume has to be really high for these guys to be released. You don't normally see these hormones being released. But if blood volume becomes really high, then obviously blood pressure is going to become high, and there's going to be lots of mechanisms that we've already talked about. Cardiac reflexes, vasomotor reflexes that are going to try to deal with that really high blood pressure. But ANP and BNP, again, are released if the atria and the ventricles are stretching due to a very high blood volume or blood pressure. And like I said, they're going to work to decrease how much water we reabsorb so that we get that water and put it in the urine, try to get it out of the blood. The other thing that ANP and BNP can do is they can cause the afferent arteriole to dilate and the efferent arterioles to constrict. So let's just think about what that would do. ANP and BNP are released if blood volume is really high, right? So they can do what we just talked about decreasing how much water gets reabsorbed to help decrease blood pressure. They can also work down here, causing dilation of the afferent arteriole, constriction of the efferent arteriole. Now, what would that do to our glomerular hydrostatic pressure? It would increase it, right? We'd ramp it up. If we're dilating this, constricting this, then blood's going to come in here. Now, at first, to me, that always seems counterintuitive, right? Our blood pressure is already high. Why do we want to make this even higher? But why would we want to make this even higher? If we make this higher, what are we going to do to our filtration rate?
Increase it.
Make it high. And if we make filtration rate high, then what are we doing ultimately to how much urine we're producing? Increasing it. And if we increase urine production, what are we ultimately going to do to our blood volume? Drop it. And then blood pressure will drop. Okay. Yeah, so don't get confused. These are happening in extreme situations. And in these situations, you want to start making a lot of urine to try to get that water out of the body.
So is that more like a positive feedback?
No, it's still negative feedback, right? So
It's just
ultimately,
ramping up faster.
the problem is high blood volume, and what you ultimately want to do is decrease blood volume. So it's still negative feedback. Anytime you maintain homeostasis, it's always a negative feedback. Okay? Now, in terms of the autonomic nervous system, again, autonomic nervous system is not going to play a big role with the kidneys. However, what have we set up to this point? If we're in a sympathetic mode, is urine production going to increase or decrease? It decreases, right? So we saw that the autonomic nervous system can influence blood pressure and things like that. But if we're in a sympathetic mode, we don't want to be processing urine, right? We want that blood to be shunted over to our skeletal muscles, and we'll deal with getting rid of those metabolic waste products later. So what will happen is sympathetic postganglionic fibers can come over and they will cause constriction of our afferent arterioles. If those guys, again, this will only happen if we're in extreme sympathetic mode. Usually the kidneys are not worrying about the autonomic nervous system. They're not worrying about hormones. They're doing their little auto-regulation. But this is if we're in extreme sympathetic mode. A bear walks in. We get mugged. So something that's really extreme, really intense, short period of time. Stop urine production. Those sympathetic fibers are going to cause the afferent arteriole to constrict. And if this constricts, what's going to happen to our glomerular hydrostatic pressure? It's going to drop. What are we going to do to our filtration rate? Drop. We stop producing urine. Again, this is only happening for short periods of time. And this is overriding those local regulatory mechanisms that are always trying to keep glomerular hydrostatic pressure and GFR constant. So this would be an extreme case where those fibers become so active that we're going to override what the kidneys normally do in terms of keeping all this constant. Now, this can become a problem for athletes. Athletes that are in endurance competitions. They will stop producing urine for, during that event. And the problem is these athletes are using their skeletal muscles, so they're producing a lot of metabolic waste. Now, usually it's okay because as soon as the event is over, you go out of this sympathetic mode, you start producing urine again, you get rid of all those waste products. That's fine. But athletes do have to look at this and be a little concerned with it. Again, usually it's okay, they do their event, these products build up, but then they can get rid of them very quickly. But you don't want to be, this is another reason you don't want to be in sympathetic mode for long periods of time because it can cause proteins to build up in your urine, it can cause all these various waste products to build up, it can lead to hydrogen ions staying in your blood too long, lots of problems, not good.
So, I mean, it's a dog, but our dog, if she runs a lot of dog park, like just sprints, she'll sprint forever, then she's like, will pee blood? And the vet said that she was just breaking her muscle tissue.
Yeah. So it's not great, right? It's not great. I guess if it's happening just a small amount, then it's okay, because it's really the same thing that's happening here. Athletes can sometimes see a little bit of blood in their urine after an event. And again, it's just because some of that will kind of start to leak through once you do have filtration. And it's because at that point, the pressure starts to get high in here once you do start to be able to form that filtrate, and then some of those cells can seep through here, or some of the hemoglobin can come in there and cause that red color. Yeah, I don't know. Short periods of time, it's probably okay. Long periods of time, probably not so great. Yeah.
Is that what happened when you were talking about the sympathetic kicking in, where you have like for extreme fear, the relief that you feel afterwards, like you're out of, you're not going to get killed or something, when you pee or you soil yourself? Soil yourself? Is that a result of all this too?
So, I'm not sure exactly the conversation you're talking about, but I think maybe at some point somebody asked about if you're really afraid, then a lot of times you will like immediately urinate or defecate, right? So what is generally happening in those situations is because of that intense fear or whatever that trauma, that immediate trauma is, if you go back and think about how defecation occurs, right? So you have to have both of those sphincters relaxed. And the same is happening for urination. You have to have both sphincters of the urethra relaxed in order for that to occur. So in extreme situations like that, any urine that's already sitting in the bladder and any fecal material that's kind of there in the sigmoid colon or the rectum or the anus, if you have extreme trauma, then those descending voluntary pathways that are keeping that skeletal muscle contracted, basically they can be overridden. So you're so concerned with that trauma, or this sympathetic flood is so intense that those pathways that normally are firing off action potentials and keeping that skeletal muscle external sphincter closed don't. And so then that can cause that immediate relief. So not so much about what's happening here, but what's going on down at the bladder and the urethra at that point. Okay. So we're going to leave filtration. We're done with filtration. Okay. And now we're over in the PCT. And again, what I want you to know about the PCT is that reabsorption is the primary thing that's going on at the PCT, and most of our reabsorption happens at the PCT. But we can also have secretion happening there as well. Secretion can happen there as well, but most of what the PCT is doing is reabsorption. So in the PCT, we're going to reabsorb about 60 to 70% of that filtrate that we just formed. We're going to reabsorb it. So we said by the time it leaves the tubule, we have to reabsorb 99% of it. So we can see that we're getting most of it done here at the PCT. Now again, what I want you to know is exactly what gets reabsorbed depends on what channels or pumps that are there. And I'm not going to ask you to learn exactly which pumps are there and how is it getting transported. Is it facilitated or is it active transport? I'm not going to ask you to know that. What all this here is, I just want you to get a sense of lots of different things are getting reabsorbed here at the PCT. So glucose, amino acids, lots of different ions are getting reabsorbed here. Water is going to be following these solutes because of osmosis. you're going to have urea, chloride, some lipid-soluble materials, and then you are going to have active secretion. You are going to have some things being secreted. Again, I'm not going to ask you details about this. I just want you to know a lot of different things are getting reabsorbed here. Now, again, don't worry about, I put this slide up here just in case you were confused about how ions are moving out of the tube and into the blood. That's what this is showing you. But I'm not going to ask you any details about that. Okay? I want you to know it in a general sense. I want to go over to the loop of Henle. Here we do want to look at some detail. So by the time that that filtrate gets to the loop of Henle, we have reabsorbed roughly half of the water, or I'm sorry, by the time that the filtrate gets to the loop of Henle, we have reabsorbed about 60 to 70% of that 99% that we need to reabsorb. The loop of Henle, here what we're going to do is, of that remaining 20 or so percent, we're going to reabsorb about half of the water and 2/3 of the sodium and chloride that we have left to reabsorb. And I'm not going to ask you those numbers, okay? Again, I want you to have a general sense of what's happening. And what's going to happen here at the loop of Henle is something called countercurrent multiplication. And people always get really bogged down and confused by this. Again, I want you to think about it in a real kind of general sense. I don't want you to worry about every nitty-gritty detail. But what this is doing, so let's look at the benefits of this, and then we'll talk about what it is. So the benefits of countercurrent multiplication is this is going to be a way to very efficiently reabsorb solutes like sodium and chloride in water before that tubular fluid or the filtrate gets to the DCT and collecting ducts, which is the last places that we have to tweak it. And again, the DCT and collecting ducts are going to be under the influence of hormones. So this is the last place, the loop of Henle, where we have kind of a setup that is reabsorbing always a certain amount of sodium and chloride in water. DCT and collecting duct, we're going to tweak that based on what hormone levels are. So having this loop of Henle is going to set up a nice concentration gradient in that interstitial fluid that's going to allow us to reabsorb water. Remember that we said if we have fluid in the tubule and we want to reabsorb the water, if we want the water to leave the tubule, go into the interstitial space, and then into the peritubular capillaries, there has to be a concentration gradient out here. This has to be high osmotic concentration for that water to move via osmosis. And this countercurrent multiplication sets up a very high osmotic gradient out here in the interstitial fluid that allows water to get reabsorbed. If we didn't have that, if the concentration gradient in the interstitial fluid was equal to what's in the filtrate, water wouldn't move. It would just stay in there. Okay, so we've got to set up a high osmotic gradient out here for water to leave and get reabsorbed. Now, this is also helping, this countercurrent multiplication is also helping to set up this osmotic gradient out here in the interstitial fluid, because as that filtrate comes over, this is the DCT here, and then this is the collecting duct. If our ADH levels are high, then we need to reabsorb water in the DCT and collecting duct. And again, if ADH levels are high, that's going to cause more water to get reabsorbed from the DCT and collecting ducts, but water will only get reabsorbed if the osmotic concentration out here is high. So what's going on at the loop of Henle is going to affect this interstitial fluid here that is going to ultimately affect how we reabsorb water from the collecting ducts in DCT into that interstitial space, then into the peritubular capillaries.
Can water pills affect that?
Water pills will definitely affect that, yeah. Okay. So what is countercurrent multiplication? The countercurrent part means that fluids are moving in the opposite direction. So if this is our loop of Henle here, this is our PCT, that means that fluids are moving down the descending limb, and then fluids are moving in the ascending limb. Fluids are moving in two directions. And because these loops are close together, what happens over here is going to affect this interstitial environment, which will then affect what happens over here. So if these two limbs were far apart, then it wouldn't matter. What happens in one limb wouldn't affect the other. But because they're close together, what's happening here is going to affect what happens over here. Now, the multiplication part, this is kind of like a positive feedback loop. We're going to keep increasing our effects based on what's happening in these two parts of the limb. So it's going to keep kind of, what happens in one limb is going to propel what happens in next, which then propels what happens to the fluid coming down next in this limb, which will then affect the fluid coming up here, which will then affect the fluid coming down in the next section, and so on and so forth. It'll add or multiply on itself. So what's happening? When you look at these limbs, they have very different permeabilities. We mentioned this before. The thin descending portion is permeable to water, but relatively impermeable to solutes. So this area over here is permeable to water. The thick ascending limb, this is going to be permeable to sodium and chloride. Now, so what's going to happen here is, okay, so the fluid is coming down here, okay, and as that, I'm going to actually start this part. So as the fluid comes up here to this ascending portion, there's little pumps in here that are going to actively transport sodium and chloride out of the fluid. So sodium and chloride are being actively transported out into the interstitial space around that ascending limb. Now, the next fluid that comes down through this descending limb, right, because then that fluid then continues on to the DCT, and now we have new fluid coming down. The fluid that's coming down here, because there's so much stuff out here in the interstitial space, the osmotic concentration out here in the interstitial space is now high. And the fact that there's a lot of stuff out here in the interstitial space is going to cause water to get reabsorbed or leave that filtrate. So that's going to help water get reabsorbed here. Now, as that filtrate comes around the bend, because all this water was getting reabsorbed, that filtrate now is going to be more or less concentrated. More concentrated. Water left the filtrate, not all of it, but some water left the filtrate. So now that filtrate is very concentrated. And as that filtrate comes up here where all those sodium and chloride pumps are, the fact that that filtrate is really concentrated means that those sodium and chloride pumps are going to be working really fast. And you're going to get a lot of sodium and chloride reabsorbed. So then the next bit of filtrate that comes down having all this sodium and chloride out here in the interstitial space is going to draw that water out really quickly. And then because that filtrate's so concentrated, these pumps are going to go to town pumping sodium and chloride out. And that's the multiplicative part. The fact that these areas are permeable to different things is going to cause these pumps to work really fast, drawing that sodium and chloride out of a concentrated filtrate. That then is going to set up a high osmotic gradient out here in the interstitial fluid, so that the new filtrate that comes down the descending limb is going to have a very high osmotic force drawing the water out. And the more water that gets drawn out, the more concentrated that filtrate is. The more concentrated the filtrate, the faster those pumps are working, so on and so forth. And this is always happening. This is a very efficient way to reabsorb a lot of sodium chloride in water, the way this is set up. Again, if these limbs were far apart from one another, if the loop of Henle was stretched way out, then what happened over here in this limb wouldn't affect how much water was being drawn out. But the fact that they're close together, all of that sodium and chloride coming out sets up a nice high osmotic gradient here, which draws that water out really well. So the way this is organized is very efficient for reabsorbing water and sodium and chloride really well. So that's about the extent that I want you to know. If you want more detail or that doesn't quite make sense, then you can start looking at osmotic concentrations of the filtrate versus the interstitial fluid, and maybe that will help you a little bit. Now, remember that the longer the loop of Henle, the deeper it extends into the medulla, the more water that it can reabsorb. And we said that humans have about 15% of those long loops of Henle's. Camels have a whole lot. And that's because, one, you have more area to reabsorb the water, but also the deeper that you go in the medulla, the more concentrated the interstitial fluid is. So if you look at the interstitial fluid down here, it's about 1,200 milliosmoles. If you look up here, it's around 300. So if you have a long loop of Henle and you're getting down into this really concentrated interstitial fluid, water is just going to fly out of that filtrate. It's going to allow for a really fast reabsorption. Okay, so then we go over to the DCT, and at this point, we only have about 20% of that initial filtrate left. And here, it is not going to look like blood plasma anymore. The concentrations of the filtrate versus the blood plasma are going to be very different. And that's because we've been reabsorbing and then secreting all along the way. Now, at the DCT and the collecting ducts, this is where we're going to make our final adjustments. We're either going to reabsorb some more material or we're going to secrete some more material. But this is the last place that we can tweak that filtrate so that we get the right solute composition and the right volume of our blood and thus of our urine. Now, the DCT and the collecting duct are under the influence of hormones. So based on what the body needs, that's where we're going to tweak things here in the DCT and the collecting duct. Up to this point, the PCT and the loop of Henle, they always do this. This is what those areas do. This is what the loop of Henle does. It reabsorbs a lot of sodium and chloride and water. That's what the loop of Henle does. Doesn't change. There's always a certain number of sodium and chloride pumps there. There's always a certain amount of water that gets reabsorbed based on how long the loop of Henle is. That's just standard, always happens. But at the DCT and collecting ducts, we can change the number of transporters, we can change the number of pumps, and that will change how much reabsorption we have of this or that. Okay. So, at the DCT, we're going to actively transport sodium and chloride We're also going to, if we're reabsorbing sodium, that means we're going to be secreting potassium. Now, the way this works here is under the influence of aldosterone. So we just review these couple hormones I want you to be familiar with and what they do here, the DCT. So aldosterone. Remember, it comes from the adrenal cortex. Aldosterone is going to be released if ACTH triggers the adrenal cortex. It's also going to be released if angiotensin II triggers the adrenal cortex. And aldosterone is going to be released if blood sodium levels start to drop, and or if blood potassium levels start to become too high. So these are all triggers for aldosterone release. Actually, I should also say too, if blood sodium levels are low, that means that blood volume is low, which means that blood pressure is low. So all of those are triggers for aldosterone release. So what aldosterone is going to do is bring these back up into homeostatic range. So the way aldosterone works is it's going to target the DCT and collecting ducts. And it's going to, remember, aldosterone is a steroid hormone or a protein-based hormone. It's a steroid hormone. So that means it can go into the nucleus and it can turn on or off genes. And the gene that it activates is the gene that encodes for the sodium, potassium, ATPase pump. So those cells of the DCT and collecting ducts, those epithelial cells, are literally going to start making more sodium, potassium, ATPase pumps. And the more of those pumps, that's going to cause an increase in sodium reabsorption. And since this is a counter-transport pump, if we're reabsorbing sodium, that means that we are going to increase excretion of potassium. Again, it's counter-transport pump. So sodium's going one way, potassium's going the other. Sodium's getting reabsorbed, potassium's getting excreted. And since water follows sodium, the more sodium that we reabsorb, the more water that we reabsorb. And the more water that we reabsorb, that's going to increase our blood volume, and that's going to increase our blood pressure. So aldosterone is working in a variety of ways. One, to regulate blood volume and blood pressure, but also to regulate the osmotic or solute concentration of the blood, in particular sodium and potassium. And that should all be reviewed for you guys from the hormone section. Now, what I also want you to know, well, actually I'll mention this. If for some reason you were to have excessive aldosterone levels, and that could be due, again, to a variety of things, but if you did have very high aldosterone levels, then you could be in jeopardy of starting to have your potassium blood levels become way too low, and that's called hypokalemia. You don't want that to happen. Obviously, you don't want your hormone levels to be excessively high, but that's one major problem with having really high aldosterone levels, is it will cause your potassium levels to really become way too low. And that can affect how your muscles are contracting. It can affect neuronal signaling. It can affect all kinds of things. It's not good. Now, the other hormone I want you to be familiar with is PTH, right? So PTH, remember the triggers for PTH. If your blood calcium levels become too low, then PTH is released. PTH is going to go to your DCT and your collecting ducts. And PTH is going to cause an increase in calcium pumps in the membrane of those cells. And that's going to cause an increase in how much calcium that we reabsorb. So the more calcium that we reabsorbed, that's going to increase our blood calcium levels. It's a job of PTH. Now, I also want you guys to know where I put it. It's in your slides, I just don't know which slide it's in, so let me just write it out. I also want you to know ADH, how ADH is working. So ADH, triggers for ADH release is going to be if your blood volume becomes too low and thus your blood pressure becomes too low, then ADH can be released. So if your blood volume is too low, or another way of thinking about that, if your blood is too concentrated, meaning that the osmotic concentration is too high. Right? Basically means you don't have enough water in your blood. So blood volume is low, or the blood is too concentrated. And I mentioned this because at one point we had said that the hypothalamus contains osmoreceptors that detect this. And it's the hypothalamus that releases, well, I should say, posterior pituitary releases ADH. It's made in the hypothalamus. And so what ADH is going to do, so ADH is going to target DCT and collecting ducts. And here it's going to cause an increase in aquaporins, water channels. So if we're making more water channels, then that means we can increase how much water we are reabsorbing. And if we reabsorb more water, that's going to increase our blood volume, and that increases our blood pressure. Remember that we mentioned that drinking actually will inhibit ADH release. It inhibits the hypothalamus from making as much ADH. And so when you're drinking, you don't reabsorb as much water because you're not making as many aquaporins. And so your urine volume becomes very high because you're reabsorbing less water. That water ends up in your urine. Now, what I didn't write on here, just keep in mind that if I say blood volume is high, then what does that mean for total urine output? It would be low, right? So on the test, I might say ADH causes an increase or decrease in urine output. And I'm assuming that you guys can take that lead from this, okay, without me writing it up there. Okay. So let's kind of go through these slides and see which are important and which aren't, because I really want to get through here. There's a lot of like kind of extra information on here, so let's just see. Okay, so what this is saying is at the DCT we can also do secretion as well. And again, I'm not going to ask you specifically what things are getting secreted. What I want you to know, we just said that aldosterone targets the DCT in collecting ducts, causes more sodium to get reabsorbed, which means more potassium is being secreted or excreted. Let me write secreted on it. So that's one thing that you should definitely know happens at the DCT. Potassium gets secreted, again, under the influence of hormones, and then other things can be secreted here too. Hydrogen ions can be secreted. I'm going to talk about that in a second. Calcium, phosphate, different compounds, drugs like penicillin, morphine, a lot of the metabolites from pharmaceuticals that we take get secreted here at the DCT. And again, this is mostly going to be from active transport. And again, I'm not going to ask you all the details. I just want you to know in general what's happening there. Now, I do want to talk about hydrogen ions because this is how we're going to regulate our blood pH. Now, this should be very intuitive for you guys. You shouldn't have to memorize this. So let's just walk through it and make sure it is intuitive. So, if your blood pH starts to become too low, blood pH starts to become too low, and that means that you have too many or too few hydrogen ions in your blood. Too many. So you have too many hydrogen ions. Okay? Too many hydrogen ions. Don't get confused by my symbol. I just mean you have a lot of hydrogen ions. If that's the case, then at the DCT, if blood pH is becoming too low, at the DCT, are you going to increase or decrease secretion of hydrogen ions into the urine? You're going to increase secretion of hydrogen ions into the filtrate, which eventually will be in the urine. Okay? And then that will cause your blood pH to go back up to homeostatic range. That should be intuitive. If the opposite were true, if your blood pH started to become too high, then that means your blood has too few hydrogen ions. And so at the DCT, you're going to decrease secretion of hydrogen ions. Okay? So hydrogen ions are always being secreted at the DCT. It's a matter of, are you going to increase or decrease how